Our last macromolecule that we're going to talk about are going to be the nucleic acids. All right. Nucleic acids are important in both prokaryotic and eukaryotic cells. In terms of our nucleic acids, we've got two types that we're going to worry about. We've got DNA and we've got RNA. That DNA is deoxyribonucleic acid, RNA is ribonucleic acid. They're going to have some things in common, all right? They're both going to have a pentose sugar that make up the nucleic acid. And I'm going to draw that sugar now, all right? And in terms of that sugar, all right, at those corners, you assume we have carbons, all right? So it being a pentose sugar means it has 1, 2, 3, 4, 5 sugars, all right? And that's the order they're arranged in, okay? You will have functional groups that will come off of the sugar, and those functional groups that come off of the sugar are going to be what tell you whether or not this molecule is RNA or whether this molecule is DNA, all right? And the carbon that's going to be important for that is going to be that second carbon, all right? We refer to that as the 2' carbon, all right? That 2' carbon is going to tell you whether or not this molecule is RNA or whether or not that molecule is DNA, all right? So I'm going to draw a hydrogen there, and I'm actually going to draw another molecule over here because I think that will be easier to see than me trying to erase something, all right? This is still our C2, C1, C3, C4, C5, okay? We have at the 3' carbon again another hydroxy group. This one I'm going to draw in a different color so that you can distinguish them. All right. So at that 2' carbon, in one of these I've drawn a hydrogen, one of them I've put a hydroxyl group. All right. If you have a hydroxyl group there, that makes this sugar a ribose. All right. So that would make that a ribonucleic acid. Okay, so in terms of that, that would be RNA. Let me put that under here. That's one way that the cell can tell the difference between something that's an RNA molecule and something that's a DNA molecule. Okay, it looks at the chemical structure. So this over here would be our deoxyribose. And this is our DNA, all right? So that's how you're going to tell, one way that you can tell RNA from DNA, all right? The next way that we can tell differences between these two molecules are going to be looking at what's attached to that first carbon. And attached to that first carbon, we're going to have a nitrogenous base, all right? And we'll have a nitrogenous base with deoxyribonucleic acid, and we'll have one with ribonucleic acid as well. There's a difference in the types of bases that are attached, though, all right? DNA is going to have adenine, thymine, cytosine, and guanine attached to it. Ribose, or RNA, is going to have adenine, uracil, cytosine, and guanine. All right? So the difference between DNA and RNA, DNA has a thymine associated with it, RNA has a uracil. So if I put up a DNA sequence for you to look at, and it has, say, ATGCC, you know that that sequence has to be DNA because of that thymine that's there. All right? if I put up a sequence that is UAG, UCC, you know that has to be RNA. And the giveaway on that is you've got a uracil there. All right? And in terms of chemical properties, the difference between having that hydroxyl group there versus the hydrogen makes a big difference in the three-dimensional structure of RNA versus DNA. All right? DNA has the ability to form a double-stranded helix. All right? And the reason that helix can form is because that hydrogen group is small. It doesn't inhibit two long chains of DNA coming together. All right? Having that hydroxyl group present in the ribose, or the RNA, inhibits two chains of RNAs from coming together. So our DNA exists as a double strand. Ribose primarily is going to be single-stranded, right? Although you will have and find some exceptions where RNA can kind of fold on itself a little bit, the chain of bases strung together are going to be single-stranded, all right? So in terms of that, we've got a nitrogenous base, we've got the sugar. Those are the two main things that you can use to distinguish DNA from RNA, all right? There's one more structure that I have left off of the DNA and the RNA molecule that I'm going to draw in now, all right? That last structure associated with a nucleotide is going to be a phosphate group. And you're going to have a phosphate group associated with DNA. We'll kind of draw it over here because I'm running out of room. You're going to have a phosphate group associated with RNA. All right? And what's key with the organization of DNA and RNA, the phosphate group is always attached to the 5' carbon. All right? The nitrogenous base is always attached to the 1' carbon, or that first carbon. Okay? So in terms of that, those are going to be important carbons to look for. Carbon number 2 is important because that tells you whether the sugar you have is deoxyribose or ribose. All right? The last location of carbons that's going to be important is going to be the 3' end. And what I'm going to do is I'm going to stop the video here, and then I'm going to redraw this to show you how that 3' end is going to become important in linking the nucleotides that we have here, because that phosphate, that sugar, and that nitrogenous base all together make up a nucleotide. All right? And we need to hook those nucleotides together to make our macromolecule, to make our chain of DNA, to make our chain of RNA. All right? So I'm going to stop here, and then I'm going to come back and show you how the DNA and the RNA is linked together.